Hello there, I'm Jim Watchorn. I'm a squadron leader and an ST7 registrar in renal medicine and intensive care medicine. And I've been invited here today to talk to you about a number of different renal topics, looking at the new renal replacement therapy capability that we're bringing in within the RAF. And my initial lecture will talk to you about renal physiology. So what are the roles of the kidney? Well, it has a number of physiological roles, but predominantly your kidney is there to maintain homeostasis in the body. Its role isn't to make urine. That's just a byproduct. Don't tell the surgeons. There's a number of processes that the kidney has which maintain its longer term homeostatic mechanisms, such as its role in bone metabolism and erythropoiesis, which are not really relevant to today's talk. They're more a problem with patients with chronic kidney disease, so I'll skip over those. And it's the other three main things that your kidney does that become a problem if you have a severe acute kidney injury. That of acid-base balance and acid-base derangement with an AKI, that of volume maintenance and your inability to maintain intravascular volume, and your inability to excrete waste, which really those three things together are the manifestations of severe AKI. So in order to achieve this, there's a number of processes which your kidney does. It can selectively maintain water, electrolytes and solutes, and in order to do this it can do it by filtration of blood, selective reabsorption of certain components of that filtrate. So there's a number of processes which your kidney does in order to keep all the good things in the blood and put all the things that you don't want in the filtrate, which will eventually become urine and be lost. And to do that, you selectively filter across your basement membrane, and then you selectively reabsorb a lot of those things that you've already filtered from the tubule back into the peritubular capillaries or back into the bloodstream. And your tubules also have an ability to secrete more substances from the blood into the tubule. So three main processes. So the anatomy of the kidney is pretty basic. I mean, you have two, they're retroperitoneal organs, and they live below your diaphragm. They have a renal artery, a renal vein, and a ureter that leave them. And the left one sits slightly higher than the right one. Supposedly, the right one's pushed down by the liver. So let's have a little bit more of a look into the kidney itself. So the diagram you can see there on the left of your screen is a cross section through the kidney, and the kidney is an encapsulated organ. Within there is, or just beneath the capsule, is the renal cortex, and in that renal cortex sit all of your glomeruli, and those glomeruluses are where filtration occurs. So that's where blood is eventually delivered through the afferent arteriole to the glomerulus. Pressure within that space pushes the blood with a driving pressure across a semi-permeable membrane, which is your glomerular basement membrane, and that drives filtration from one side, from that plasma blood space, down into that Bowman's capsule, that bit beneath where you're starting to generate filtrate. So all those little filter mechanisms, the glomeruli, sit in the renal cortex, which is the outer layer. Then as you go a bit further down within the kidney towards the centre of it, you get into the renal medulla. And there are two components of the renal medulla, the outer and the inner medulla, which you can kind of, for the sake of today, think of as one bit. They look slightly different if you were to actually cut through them. And in the renal medulla is the concentrating engines of your kidney, your loop of Henle. They sit there, and also through that medulla passes your collecting duct, which takes your eventual filtrate, which has become urine by the time it leaves the nephron, collecting duct down to join the papillas, which eventually join together in the renal pelvis and pass down into the ureter and take urine to your bladder. So in the medulla, loop of Henle, collecting ducts, and you also have arteries and veins transiting through to the renal cortex. So now we're going to look in a little bit more depth at the individual components of a nephron. And a nephron is one single urine-making apparatus, really. So it describes the cellular apparatus from blood side to urine side. So if you look closely at my diagram, so now we're going to look at the diagram on the right. And if you look closely there are two nephrons. There's a nephron on the left, and you can see the glomerulus, and there's a nephron on the right. The difference is one has a deep looped loop of Henle and one has a shallow loop of Henle. Otherwise they're essentially the same, and they join on that common collecting duct down the middle. So in the glomerulus, that's where blood arrives at the filter mechanism, and it comes in through the afferent arteriole, and once it's filtered across, it passes into the proximal tubule, and we'll look at each bit in a bit more detail shortly. The proximal tubule then reabsorbs an awful lot of stuff that's been filtered, goes down into your loop of Henle, which is your concentrating mechanism, and then into your distal tubule, and you have a distal convoluted tubule, and it eventually pass into your collecting duct. The difference between the shallow and the deeper loop of Henle really dictates how capable that nephron is at concentrating the filtrate. The deeper down you go into the medulla, the higher the osmolality, the higher the concentrating mechanism within that segment of the loop of Henle. So let's look in slightly more detail at the glomerulus. So as I've said, blood arrives in the afferent arteriole, and then it's driven by a pressure across the filtration membrane, which is predominantly glomerular basement membrane, but on both sides you're going to have capillary endothelium. So the afferent arteriole has got specialized lining to allow fluid to leave the vessel. It then, and that sort of gaps there of around sort of 100 nanometers, so most stuff can quite easily pass straight out of the capillary tube, straight out of the capillary at that point. But then you go through the glomerular basement membrane itself, which is this three-layered structure, which is much more selective and retains most things, down to the podocytes. And the podocytes make up the lining of Bowman's capsule, and the foot processes of the podocytes have specialized molecules on them, which are charged, which really narrow down the size of the molecules that it allows to pass through. So things like water, urea, potassium, they're all really small molecules. They can easily pass through. But albumin, for example, is a larger negatively charged molecule, and the podocytes themselves are negatively charged, so it totally repels albumin and keeps it within the capillary, and it's not filtered whatsoever in health. So let's look in slightly more detail at the glomerulus. So as I've said, blood arrives in the afferent arteriole, and then it's driven by a pressure across the filtration membrane, which is predominantly glomerular basement membrane, but on both sides you're going to have capillary endothelium. So the afferent arteriole has got specialized lining to allow fluid to leave the vessel. It then, and that sort of gaps there of around sort of 100 nanometers, so most stuff can quite easily pass straight out of the capillary tube, straight out of the capillary at that point. But then you go through the glomerular basement membrane itself, which is this three-layered structure, which is much more selective and retains most things, down to the podocytes. And the podocytes make up the lining of Bowman's capsule, and the foot processes of the podocytes have specialized molecules on them, which are charged, which really narrow down the size of the molecules that it allows to pass through. So things like water, urea, potassium, they're all really small molecules. They can easily pass through. But albumin, for example, is a larger negatively charged molecule, and the podocytes themselves are negatively charged, so it totally repels albumin and keeps it within the capillary, and it's not filtered whatsoever in health. So what is driving that pressure? And as most of you anaesthetists, you do love a pressure equation, and you have a driving pressure on one side in the capillary, which is driving fluid and generating filtrate. It's pushing fluid from one side to the other. And so that driving pressure is a combination of hydrostatic pressures pushing and oncotic pressures pulling, or osmotic pressures allowing passage through. So you have a driving hydrostatic pressure in the blood, which is that P, little h for hydrostatic, little b for blood. But pushing back against that hydrostatic pressure from the capillary side is the pressure, which is hydrostatic, but in the filtrate. So there'll be a little bit of pressure in that pushing against it. And it's the balance of those two. And that's offset against the pulling pressure. And that's the oncotic pressure, which wants to hold fluid in the capillary, offset against the oncotic pressure in the filtrate, or in Bowman's capsule, wants to hold fluid in Bowman's capsule. So it's the driving the hydrostatic pressures minus the oncotic pressures, which gives you that filtration pressure. And that generates your what we call single nephron glomerular filtration rate. Is that driving pressure across? And then if you multiply the amount of nephrons you've got in health, it's about 1 million in each kidney, so times 2 million, it gives you a overall glomerular filtration rate of about 120 mils a minute per metre squared, which is the typical units that we quote GFR in. So most people in health, GFR is around 120, but it's age, sex and race dependent. Now, if a molecule moves from one side, the capillary side, into the filtrate side entirely and is freely filtered and it's not reabsorbed, then your clearance of that molecule from the plasma is equal to your glomerular filtration rate because they're the same. So yeah, clearance equals GFR. And that is roughly true of a molecule like creatinine, which is why we use creatinine to give us a measure of renal function. But it's not entirely true because creatinine is also secreted by the tubules, which makes it slightly more complicated as opposed to a true measure of something, say, like inulin, which we use, was in the textbooks as the gold standard measure of GFR. So that's how we measure GFR. And obviously renal plasma flow is something different because not all of the renal blood flow gets filtered. A lot of it leaves via the efferent arteriole and supplies round the rest of the blood supply, leaving the efferent arteriole, which is still arterial, and it goes into the vasa recta, which is the blood supply to the loop of Henle and the rest of the nephron. And so your renal plasma flow, your renal blood flow, is about a quarter of your cardiac output, because you've got two kidneys, about 650 mils a minute in each kidney, but your GFR is only 120 mils a minute. And throughout a range of different driving pressures and blood deliveries to your kidney maintains an ability to keep its glomerular filtration rate, its GFR, exactly the same. And that's through two main processes, but both of those main processes cause auto-regulation of your renal blood flow. And I think you see this quite frequently, you talk about it quite frequently in anaesthetics because you are at risk of having hypotension in theatre. But across a range of blood pressures, essentially, your kidney has an ability to maintain the same GFR, and it does that by selective changes in your afferent and efferent arterioles. By two reflexes. One is myogenic, so the actual afferent and efferent arterioles sense a change in pressure, and to increase GFR, so if pressure drops, for example, the efferent's going to squeeze up and get a bit narrower, and the afferent's going to dilate, get a bit wider, and you can imagine then you're increasing the pressure that's driving fluid across the basement membrane. You're increasing or maintaining GFR despite that drop in blood pressure. And the other way you do it is by sensing the amount of sodium that's delivered into the tubules through your juxtaglomerular apparatus, and those sodium sensing cells in the tubules feed that back straight to the afferent and efferent arterioles. So they're the bits that control your autoregulation, and that permits you to hold the same GFR no matter what blood pressure you have, provided it's within a certain range. And once you get outside that range, so if you become particularly hyper or hypotensive, blood pressure is sort of less than maps of 70, 60, then you lose that ability to autoregulate and your GFR drops because your kidney can only cope so much. So how does your kidney take an awful lot of filtered fluid, that 120 mils a minute of fluid? Imagine if you generated that and you had no way to selectively retain it, it wouldn't take very long for your entire circulating volume to be pushed into filtrate. How does your kidney take that filtrate and turn it into a small amount of urine? And it's done by selectively maintain a number of specific molecules that you don't want to get rid of, things that you really want to hang on to. For example, calcium, magnesium, phosphate, all those things that you kind of want to regulate really closely, but also get rid of all the things you don't really want, like potassium. There's two main processes. They both transport across the tubules. One is passive, and that passive movement could be simple diffusion. That's how water usually is retained. And facilitated diffusion, so that's through selective pores for things like sodium. And then you can actively transport, so this uses ATP to generate a driving gradient, and it's pushing against a concentration gradient, and that's why you would use active transport, because you're pushing from a already low concentration to a high concentration on the other side. So we've looked at the glomerulus in some detail and the way you create filtrate. Now we're going to look at the individual parts of the nephron further down. So as you can imagine, 120 mils a minute passes into your proximal tubule, which is a vast amount of fluid. And you want to draw most of that back from one side, from the tubular side, back into the capillary. And a lot of that work is done passively by your proximal tubule. Your proximal tubule receives about 180 litres of plasma per day into it. And it's a bulk reabsorber. It drags most of those things back. To do that, it has a sort of a convoluted surface. It has villi within it. And it also has lots of mitochondria because it's very active, pumping, with some active transport, pumping some of the things back that you want to keep back. It has a convoluted part to it, so a looped part and a straight part, the convoluted and the rector. And this is where you pretty much reabsorb all of your glucose that you don't want to pass into your urine, your amino acids, your low molecular weight proteins, all those things that you want to get back and maintain in your plasma. And about 65% of that filtered water is put back into the plasma space by the proximal tubule. It's isoosmotic, so there's not a lot of active pushing against gradients going on here. It has leaky junctions, which allows water to pass with everything. And it also has an ability to get rid of some weak acids, like ammonia and things that you don't really want. So we've looked in some depth at the glomerulus and the proximal tubule. And now we're going to look at the loop of Henle. And the purpose of the loop of Henle, what it does is it generates that osmotic gradient of your kidney. It's the concentrating gradient, or the engine, that allows the rest of the processes in your kidney to occur. So let's have a look at this top diagram here, and we'll pretend for a minute that the loop of Henle doesn't exist and your kidney is just trying its hardest to generate a concentration gradient. So we've talked about fluid coming in from the proximal tubule, which comes in at this end. And that's isoosmolar, because nothing's really concentrated, nothing's really altered the difference between solutes and water, got rid of one or kept one and got rid of the other. So the osmolality coming in at the top of your loop of Henle is going to be that of the blood. And for today's lesson, we're going to call it 290 milliosmoles per litre. So it arrives at 290, and now what your kidney or your nephron does is want to pump out salt and get rid of water. Maintain some of your sodium, but be able to get rid of water. So as it comes along here, it's going to be 290. As it goes into the thin part of the tube, loop of Henle, it'll be 290, and then it could be 290, and we go from the water permeable side, which is the thin side, to the water impermeable side, which is the thick side. So if we were to pump sodium out at this point, we would pump sodium out, but water would follow because it's permeable to water. But if we got to here and we pump sodium out, because it's thick and impermeable to water, water can't pass. So there'd be no water leaving through here. And in that scenario, your osmolality would go down. So if you pump the sodium out, your osmolality will go down if water can't follow. It becomes more dilute because the solute's been removed, but the water can't follow it. So the osmolality could drop to, say, 270, because these little engines that are pumping out, the sodium pumps, what's called the sodium, potassium, ATPase, which are the engines there, they pump out the sodium, but the water's not following, which generates a concentration across it of 270. But the engines can only do so much. So if your loop of Henle didn't exist and it was straight, all you could do would be to change your osmolality a little bit to make it more hypoosmolar, but by a shift of about 20. So we've looked in a bit of detail at how the glomerulus works and how the proximal tubule works. Now we're going to look at the concentrating mechanism of your kidney, your loop of Henle. So I'll bring you over here to the whiteboard. As we've said, that the proximal tubule is a mass reabsorber, but there's not a lot of active process going on. Water and the solutes haven't dissociated from each other, so there's no change in the concentration of the filtrate as it leaves the proximal tubule and goes into the loop of Henle. So the fluid arriving at the beginning of the loop of Henle will have the same osmolality as the blood, because nothing's acted to drive concentrating mechanisms one way or the other. So let's say that the osmolality coming in here is the same as the blood, and we'll call it 290 milliosmoles per kilo. That drives in through the start of the loop of Henle. And let's pretend that you don't have a loop of Henle. All you have is a tubule that is trying its hardest to concentrate urine without the loop. So fluid comes in at this side and it's concentrated at 290, and it goes through the thin part to begin with, which is permeable to water. So if in the thin part we were to drive out sodium, water would follow, and as a result the concentration would be unchanged. It will remain 290 milliosmoles as it enters there. What we really want to do is pump out sodium but hold water back in the tubule. We want to get rid of excess water, but you need your sodium and you want to hang on to it so you can selectively reabsorb it later. So what we need are some cells that are tightly packed together, that have sodium pumps on, that can pump out the sodium, but the water stays within the tubule. And for that, you need the thick limb, okay? And that's what we represent over here. So if we had some cells here that pumped out sodium, then we would find, as a result, we would get no water passage. Water wouldn't be able to follow. But those little pumps which you have can only do so much. So they can usually generate a gradient of about 200 milliosmoles. So it could drive it down. If we drive out all the sodium and we keep all the water, we might get our osmolality in the tubule down to, say, more or less 100 milliosmoles per kilo. But we couldn't do it any further than that, so we'd drive out the sodium, we wouldn't get a lot of water follow, and we would end up with a lot of sodium left in here and a lot of water left in here. But we really want to hang on to that sodium. So your body has played a clever trick in order to do this by taking the loop of Henle, or this tubule here, and turning it into a loop by dragging this bit downwards. And I'll just bring the camera down to the other diagram at the bottom. So now we're looking at what you actually have, which is the loop of Henle. And this is your concentrating mechanism. But the same pumps still exist in this as they did in the first diagram. So here, in the thick ascending limb, it is impermeable to water. Water cannot pass. But there are lots of engines in here that are driving out sodium. So if we were 290 here, as it goes down, don't forget salt and water pass freely in the thin descending limb. So that could pass out. So it's still going to be 290 as it arrives down here. And it will be 290 as it arrives over here. And then you're going to pump out some sodium. And so the osmolality will go down to, say, 100. That's nothing new, and it's what we've already talked about at the top. But if we pump out sodium here into the interstitial space, salt and water can follow, or water particularly can follow, because water always follows the movement of sodium, and vice versa. So if sodium gets pumped out here, water will leave here. So sodium gets pumped out, but water can't follow, and the osmolality is dropped. But water can leave in the descending limb. So water leaves here, and hang on, we've just removed water. So the concentration in this part will increase. So instead of it being 290 down here now, if we pumped it out by 200, we're going to increase it by 200. So let's say it goes up to 500 for round units. So the osmolality is now 500. So the osmolality arriving at the beginning of the thick ascending limb isn't 290 anymore, it's now 500. And that's going to meet our sodium pump here, which is going to pump out more sodium. So let's take it up here, and we're going to pump out a bit more sodium. And if we pump out a bit more sodium, a bit more water wants to follow. And if we pump out a bit more water here, let's say now this arrived at 290, now it's arriving at 500. So this down here is no longer 500. This down here is now gone up by another 200, and that's now 700. So it's even more concentrated. And as you come down here, what's delivered to the thick ascending limb is no more 500, it's now 700. And that will come up here. And let's pump out a bit more sodium higher up here. So we pump out a bit more sodium. Water wants to follow, which it will higher up. So we're going to pump out water. Oh, we're not going to, sorry, water's just going to passively follow. We're not pumping water out. It follows the sodium on the other side. So now it arrived at 290. Down here it's 500. Down here it's 700. And down here it's 900. And so it's arriving at 900. And your kidney has an ability to do this until it gets down to a concentration down in the deep part of the loop that is 1200 milliosmoles per litre. Milliosmoles per kilo. We'll forget about the units. And that's how your kidney, by becoming a loop, generates this concentrating driving gradient, removing sodium on one side and water passively following on the other. So where your glomeruli at the top, this has the same osmolality of blood, but down here at the bottom of the deep loop, in the deep medulla, you have an osmolality that reaches about 1200 milliosmoles per kilo. Okay, so we're back from the whiteboard and we're looking at the lecture again now. And we talked about what really is the point of the loop of Henle, and that's to generate a concentration gradient from the interstitium to the tubular space, which you can then selectively reabsorb against, because those sodium-potassium pumps, that are energy hungry with loads of mitochondria in a thick ascending limb. They're your engines. They're the bits that drag out all that sodium, put it into the interstitial space and generate a concentration gradient from one side to the other. You can then selectively reabsorb against, because you've done the active bit in the loop of Henle. And it's done by that unique loop that we've discussed. And it generates that concentration gradient up to 1200 milliosmoles, and it's the point at which you reabsorb about half of your sodium, and that's through that active reabsorption in a thick ascending limb, which is water impermeable. And so we deliver from that a hypotonic fluid to the distal tubule, because those pumps have generated a gradient across, and the fluid leaving the distal tubule is hypotonic, but we've retained most of our salt and water in this, and it gives us that concentration gradient to selectively reabsorb against. Its blood supply is through, again, a mirrored loop of blood supply, so it looks like the loop of Henle, it wraps around it, and that's called the vasorector, and that allows passive exchange between, because it concentrates as it goes down into the deep medulla and then dilutes as it comes out again. And now we're going to move on to talk about the distal nephron. So you've set up that concentration gradient and you're delivering it to the distal nephron, and this is the point of your selective sodium reabsorption. So here in the distal convoluted tubule is the point at which you can reabsorb more or less sodium and water as a result of whether you are overloaded or dry or hyperosmolar or hypoosmolar. There's two main cells in this area. One's called the principal cell, which is part that aldosterone works on, which is active in active sodium retention and potassium excretion. And you've got your intercalated cells in this area as well, which maintain your acid-base balance, some of which pump out hydrogen, some of which actively make bicarb. We're just going to look now at sodium reabsorption and where it occurs. So the majority of it's reabsorbed by the mass reabsorbing part of your nephron, which is the proximal tubule, and that's passive. And then you end up, so let's say that's about 50%, you end up with about another 40% being removed by your loop of Henle, and it's a sort of more closely associated structures, leaving about 10% to pass into your distal convoluted tubule for selective reabsorption, and that's under the hormonal control of aldosterone. And in typical circumstances, you will then retain nearly all of your sodium, so only about 1% in health will get lost into your urine, because you want to hang on to that. So yeah, of the 100%, not very much gets wasted. Whereas if we look at potassium, you want to remove potassium in your urine. So again, a similar amount is removed by the proximal tubule. None is really removed by the loop of Henle, and then you have active movement of it again by aldosterone, which occurs further on in the distal collecting duct. And you can really get rid of between 1 and 100% of your potassium, depending on what your kidney is sensing through its homeostatic sensors. So how do you control water and how much water you release in urine and therefore keep back in your plasma or withheld by your kidney? And the predominant hormone that keeps that back is vasopressin. As you remember, as you go down into the deep medulla, you've generated a massive concentration gradient where it gets up to about 1200 milliosmoles. Vasopressin, which is released by the hypothalamus and through the pituitary, inserts little gateways into the collecting duct and the distal tubule, which allow water to pass, because normally this part is impermeable to water. Vasopressin allows those little channels to appear, those aquaporins to appear, and water really wants to leave from your tubule, which is around 100 milliosmoles, into your interstitial space down in the deep part of the medulla, 1200, a massive concentration gradient. All you need to do is selectively put in a little pore, which is going to allow water to move through it, a water-specific gateway, and water will flood out of your collecting ducts and your distal tubules and into that interstitial space and be recycled. And vasopressin is sensed and released by your osmoreceptors within the brain, and they allow this to occur. So that's vasopressin. So vasopressin is there to maintain your osmolality of your blood. It's there to maintain the concentration of your blood to its solutes. But now we're going to look at the hormones that control the circulating volume. And this is done by the renin, aldosterone, angiotensin axis. So your kidneys have within them, which we've already touched on, sensors which know how much volume is being delivered, which is through juxtaglomerular apparatus. So they sense this drop in blood pressure, as does your myogenic reflex of your afferent arterioles, as we've already talked about. And they release renin. And renin is an enzyme which converts angiotensinogen to angiotensin 1, which is further converted to angiotensin 2 within the lungs. So angiotensin 2 has multiple different roles within the body. Firstly, it can act directly to reduce GFR by constricting the blood supply that supplies the glomeruli, and therefore GFR drops as a result of angiotensin release, which pushes up or maintains your circulating volume because you're not filtering or making so much urine. So it does that. It has reactions with your sympathetic nervous system as well, and it also acts on the adrenal glands to promote aldosterone synthesis and aldosterone release. Aldosterone then comes down and it acts directly within your kidney, within those tubule cells that we've talked about that are in the distal convoluted tubule, and they actively retain sodium at the expense of getting rid of potassium, and the water follows sodium through those special cells in the convoluted tubule, in the distal convoluted tubule, to maintain your circulating volume, but you get rid of potassium as a result, which is why if you gave an aldosterone antagonist such as spironolactone, you will inhibit that, drive up potassium because you're not getting rid of it anymore because you've blocked it, which is why they're potassium sparing diuretics. Okay, and that's the end, you'll be pleased to hear, of your renal physiology lecture.